Soft X-ray tomography. We're doing it up at the Lawrence Berkeley Lab at the synchrotron. I want to give you a bit of information about how it works and what the data contains. So, let's see. First of all, we're looking at whole hydrated cells in the native state. They're cryoimmobilized. There's no chemical fixes. And it's natural contrast, no stains, and it's quantitative. So it's about as close to natural look at the structures as you can see. It's very much like a CT scan of your head or your knee. You put a cell, you put the patient into a machine, take images from all the way around the patient, numerous angles, reconstruct it. Then you get to see what's inside the patient without having to cut them open. So it's the same analogy that we're going to use looking at cells. And to get some molecular information, we use correlated imaging, looking at a fluorescent molecule superimposed on top of the structure from the X-ray microscope. So we see structures with X-rays and molecules with fluorescence. It's a full field transmission X-ray microscope, not a scanning. The light source is the synchrotron light source, radiation about 547 eV, so it's very soft, low energy. Soft means you're using very low energy X-rays, nowhere close to what it takes to do an X-ray of a human body. It's about a 2.4 nanometer wavelength, so technically that's theoretical resolution. We're far from theoretical resolution due to the optics. The optics are zone plates. They're nanofabricated Fresnel lenses. And the contrast mechanism is absorption. So we're absorbing the X-rays. The more X-rays it absorbs, the higher the contrast. This is a picture of the instrument. It's about 75 feet long. The only region that the user sees is this portion out here, right here, where the microscope itself sits. It's a very simple microscope. The schematics show you that it's a light source, a bend magnet light source. The condenser zone plate focuses the source onto the specimen, and the specimen is sitting right there. And the objective zone plate magnifies the object onto the CCD camera. Very simple, 2 lenses, specimen, and a light source. Just happens to take a complex synchrotron to get that energy of X-rays. The really cool thing is the contrast mechanism. That's what makes this exciting to me as a cell biologist. What we're doing is we're imaging between the K-shell absorption edges of carbon and oxygen, right in that sweet spot of 517 eV. And what that means is our body, carbons, proteins and fats and carbohydrates are loaded with carbon. That's what they're made of. So all of life contains carbon, which means we can look at any biological subject of interest. And it's quantitative. The absorption is linear with respect to thickness and concentration. So the more carbon per voxel, the more molecules per voxel, the higher the contrast. And you can see that with this increasing concentration of molecules in these capillaries. And so that gives us a measure, the linear absorption coefficient measurement for each region of the cell. So what you can see here is a cell, this is the nucleus, has a lot of heterochromatin, very highly compacted heterochromatin around the periphery, so it's highly absorbing. And mitochondria have a lot of carbon and membranes. They have cristae that cross the mitochondria. So a lot of carbon per voxel and high contrast. And lipid drops, of course, which are predominantly carbon, are very high contrast. So you get measurements that help with some of the segmentation that we like to do. So how do we do this? We put cells in a glass capillary, which we pull, very thin-walled glass capillary, so it doesn't absorb much of the X-rays, rapidly freeze them, put them into the microscope, and then collect the tomographic data set. You push one button, and all the images are collected from around the cell, and you can do that all the way around the cell, 360 degrees. Data are instantly sent up to our computer. Axel wrote a wonderful program that automatically aligns all of the images and does a reconstruction, and within 2 minutes, you have the entire tomographic reconstruction back in front of you at the computer. Now, when we first started doing this, it used to take us 3 hours to align one cell. So that restricted the number of cells we could get. Now, obviously, we can produce a lot of cells. Why do we use this glass capillary? Why do we want this full rotation tomography? Well, that's because if you put them on a flat substrate, as you tilt, there's a certain region that you can no longer image through. It becomes way too thick. You can see that it becomes 3 times as thick as it was. And all of a sudden, you just simply can't image because you've got to glass slide or a cover slip that's perpendicular, blocking all of your imaging. So this causes what's known as the limited tilt missing wedge problem that's very common to cryo-EM. We don't have that problem because we use this glass capillary. You can get an example of what that missing wedge causes. If you look at this issue here of decreasing the rotation angle and the loss of information as you do that, especially in the XZ orientation. Now, the cryo-EM people have come up with algorithms to retrieve that information, but we don't have to do that. We just get the full rotation and don't have to worry about missing wedge. And this is the kind of data set we see. So these are optical sections. We're just flipping through the computer data set and looking at the entire cell. You can see the nucleus, endoplasmic reticulum, mitochondria, all sorts of lipid drops and autophagosomes and nucleoli inside of the nucleus. And so you get the entire cell right in front of you. And this becomes a real advantage because there's a lot of cell heterogeneity. There are no two cells that look alike in nature, in biology, in the organism. You can take a dish of cells. That was one cell from the dish. You look at the next cell, and it will be very different. You'll see it looks more polarized. You got the nucleus at one end, organelles at the other, and these layers of endoplasmic reticulum down at this end. It's a polarized cell. The nucleus is completely round in this cell. It wasn't in the last cell. So you can't average like you can with a protein. You can't do protein averaging. You'll end up with the lowest resolution you could possibly imagine of a cell. So we have to do numbers and try to get statistical information and make sense of the data that we're collecting. Now, the next problem we have is using those data that we have, and that requires this segmentation, where you outline organelles of interest so that you can get one specific, say, measurement of mitochondria, all the mitochondria in the cell, et cetera. And that segmentation is right now very tedious, and we're hoping to get help from people like you to make it much less tedious and more automatic. That also makes it more reliable. You remove that subjective human input. So what can we do to date? What have we been doing to segment structures? Well, we first started off by just manually segmenting the nucleus, tracing around the nucleus in an image processing program that we call AMIRA. It's commercially available. You probably know it. And then you grab all the voxels that were in that nucleus, and you can plot the voxels of the nucleus here in the green and blue, and the cytoplasm in the background. I'm particularly interested in the nucleus, which is why I'm showing this here, but it allows you to see two regions of the nucleus, a bimodal curve. And there's the most normal nuclei have a bimodal curve. You can then, in that image processing program, adjust the histogram to highlight the interesting area. And what we've done here is highlight the high contrast region, which according to old EM has been called heterochromatin or silenced genes. You can highlight all of that, and then the remaining region is the euchromatin region, which is where all the active genes are located. And then you realize that it corresponds to this very highly absorbing peak here, and then you can get the 3D volume of it. So now you can have a measurement of the total volume of all the heterochromatin and the total volume of all the euchromatin. And there are biological reasons why that's important. So that's one thing we can do with the histogram segmentation. You can also grab all the mitochondria. They have a linear absorption coefficient in this range, and you can visually see that on your screen as you're adjusting the histogram to make sure you have the organelle of interest. Then you can grab all of those voxels, and now you can see the distribution of all the mitochondria in around the cell. And what it tells you is the total volume of mitochondria. That's important for things like diseases. There are changes in mitochondrial volume in cancer, in diabetes, in all sorts of diseases. So if you can get information about total volume of mitochondria, that's very helpful. And you can also get spatial distribution of those mitochondria around the nucleus and throughout the cytoplasm. And that changes with various biological activities. So to be able to grab this information automatically without having to do this manual segmentation even, the histogram adjustment, would be extremely helpful. It gives us information that tells us how crowded regions are, for example. To find out what it really means, what do those linear absorption coefficients mean? How crowded is something? Well, it made a yeast express an alcohol oxidase crystal, and we could calculate what the linear absorption coefficient could be because we know the composition of that alcohol oxidase. And we calculated the linear absorption coefficient. It came out almost precisely what we were measuring at 0.62. And that tells us that nothing in the cell is as crowded as that alcohol oxidase crystal or any crystal, except you can see a very thin line out here, which is the lipid drops. And the lipid droplets can be equally as crowded. Sperm chromatin, which is the most crowded chromatin in any cell, is about the same absorption coefficient as some of the lipid drops. So this just gives you a clue for how crowded things are in the cell. And some people, I have a friend on the East Coast, Sam Isaacson, who's a mathematician. He models how molecules move through the nucleus, for example, based on those linear absorption coefficient measurements, because he can tell how crowded it is. And he actually published that paper. I should have put it in this talk. I apologize, I didn't. But he's also using these measurements to calculate how molecules can move through the cytoplasm. So it tells us that heterochromatin is about 30% more compacted than euchromatin, and sperm chromatin is about 54% more compacted or crowded than heterochromatin. So we can get measurements like that that you can't get from other technologies. And why do we want segmentation automated? Well, here's a cell that we've segmented the heterochromatin, euchromatin, mitochondria, and the one Golgi using the histogram adjustment. But then on the return trip through that cell, we're going to see the endoplasmic reticulum, this network in red. It is so complex, so difficult, we've manually traced it in maybe 3 cells. It's really extremely difficult to segment and would drive people crazy if you tried to do that routinely. But it's an extremely important measurement to have, the total volume of endoplasmic reticulum. So we need to be able to grab all of the structures. Now that's just 4 organelles in a cell. There are a heck of a lot more organelles in a cell that we haven't started segmenting yet. So we have a huge problem, and we want to be able to get more models like this, but even fill in more of the details. Now, there's been some attempts to do some of the automated segmentation, and some of it was done here on campus with Jamie Sethian's lab and Daniel Pelt, who was here as a postdoc for a while. They did some machine learning on, I think they used 7 cells that we had manually segmented for heterochromatin, euchromatin, lipid drops, and mitochondria. And they trained the computer to learn those structures in 7 cells. And then we're able to go back and machine segment this cell. And you can see that it's almost identical. And whether the machine is more accurate than the human, I don't know. But those are things that we need to find out. Now, this isn't as robust as we would like. And let me just show you the 3D. A little hard to see in this lighting. Colors are hard to see in lighting. But it's not quite as robust as we need. And I think Axel's still working on tools to make it more robust, as is Daniel and Jamie. And other people around the country are trying to do some machine learning to grab all the information we have in these data sets. Tell you a little bit to try and convince you that it is important that we need to know these things, that it's going to provide valuable information if you can make these algorithms. One of the things that we can do is look at chromatin condensation as blood cells differentiate so that we can learn what's going on as they de-differentiate, causing cancer, causing other diseases, because that's what happens with cancers. In differentiation, cells are becoming silenced, genes are becoming silenced, the nucleus changes. In cancer, they abnormally become active. Certain genes abnormally become active. So with this tool, we can, using that manual segmentation or the histogram segmentation, we automatically segmented the nucleus, the heterochromatin and euchromatin of cells during the process of blood cell formation. We use cells from mice because they're normal and you're getting real data out of them rather than cultured cells. And you have to do it to get a differentiating cell anyway. You have to get cells at different stages of differentiation from the mouse. So these were from mouse done with Camilla Forsberg's lab at Santa Cruz. And this is just a brief view of hematopoiesis where you're taking a multi-potential stem cell. This is not your embryonic stem cell. That's what the hematopoietic is, one that can only become blood cells. It has 2 choices. It can become a myeloid or a lymphoid cell. These are your white cells down here, basophil, neutrophils, these sorts of things that protect you from foreign objects, or T cell and B cell that are part of your immune system that attacks viruses and germs like that. These guys get the bacteria, these guys get the viruses. And they also cause a lot of problems when they don't function properly. So here's what the multi-potential cell looks like. the hematopoietic stem cell, and here's what the myeloid progenitor cell looks like. So it's slowly differentiating. It can only become this group of cells here. What you can see is that you're getting an increased band of heterochromatin around the periphery, and the cell nucleus is getting smaller. You can look at the lymphoid progenitor. It's doing much the same thing. They have much more heterochromatin around the periphery. Then you can look at the neutrophil, which is a fully differentiated cell here, and you can see it has a whole lot of this blue heterochromatin, very thick band of blue heterochromatin around the periphery, as does the B lymphocyte. We can quantify that. These are just one single view of it, but you can quantify it, and we've done that here, and you can see that the percent euchromatin increases from 24% to 60%, which means that the percent heterochromatin is increasing, which means that the genes are turning off. These are the active genes. They're turning off, and you get more and more of that heterochromatin. So that's a quantitative measure which has, for the first time, quantified that this process is occurring during hematopoiesis. And it seems like most cells in the body, as the cells differentiate, you get that band of heterochromatin around the periphery. And it's important that they do that. But there are some cells in the body that do it differently, and that's the olfactory epithelial cells. And so we looked at some cells during neurogenesis in mouse, and this is when the cells are turning off a lot of genes and allowing one to just be expressed. So these cells have something like 1,400 genes per cell for olfaction. But the differentiated cell expresses only one of those 1,400. So it goes through this elaborate process of turning off all but one gene. And each cell turns on a different gene. So it's highly orchestrated. I don't have time to show you the whole story, but I want to show you how you can get information just from the X-ray images and why we need automated segmentation. We got cells at the different stages of development, and we could identify them because they each had a different fluorescent tag. And so you can pull them out in a fax machine based on their fluorescent tag, which would tell you what stage of development they're at. This, again, is from mouse tissue, from the little nose. Then you can look at the distribution from the stem cell to the maturing cell and the fully differentiated cell here. And you can see that even mitochondria goes through a reorganization, and the entire nucleus and cell are reorganized. But you can quantify that, and you can see that in this cell, rather than heterochromatin going to the periphery, it goes to the center. And that's critically important for their normal differentiation. The bimodal curve telling you that the percent heterochromatin increases from 33% to 41%, and the spatial distribution showing that massive heterochromatin in the center. And then you can quantify that, the distance from the nuclear envelope inward of the euchromatin and the heterochromatin. So here you get quantitative information, and right now biology, cell biology, is trying very much to turn quantitative. So being able to get this kind of information is very important in helping move towards quantitative rather than just visual pictures. You can get a lot of information. Spatial distribution is great. You can get that very nicely from these pictures, but you need to be able to segment that information out and get the quantitative to be able to make a statement. Because again, none of these two cells, none of the cells in this population were identical. You have to average, and we did 30 cells for each of these measurements to get a significant measurement of the change in heterochromatin. Now a little bit about the power of 3D imaging. Why does tomography matter? One little example, the old data showed that from TEM, because they were looking at a 2D slice, just one slice, they used to say there are islands of heterochromatin, just these little islands. Well, if you follow that heterochromatin in this cell, follow the blue, follow the green, you'll see there are no islands. They're continuous networks. Pixel analogy, voxel analogy, 99% of the voxels are connected. They are not isolated. If you do a skeletonization, so that old theory is debunked. If you do a skeletonization, just shrink it proportionally, you can see that there's a backbone network. And this is an Amira program too. And you can see that that network, even though the heterochromatin and euchromatin are moving around, the network remains continuous. So there's some need for that continuity. We don't know what that is yet, but we have to try to understand it. And every cell, every nucleus we've looked at has the continuity. We haven't been able to break it yet. I'm still looking for something to break it. So that's a little bit of 3D volume. More nucleus, you can look at bacteria and look at the nucleoid and see the different regions, supercoiled domain here in lag phase, in log phase, and in the stationary phase. So just examples of kinds of things you need, and each case you need statistics. A lab on campus, Chris Mayogi's lab, looking at algae, bioenergy information. This is a single-cell algae. It grows and multiplies and turns into a 16-cell organism. All of these structures have been manually segmented out. This is a complete manual study, which took months. And what you can see is the different structures within organelles that are identified here don't really matter. It just shows that from this information, you can look at a normal control cell, 16-cell stage, and then you can look at manipulation of glucose to try and increase the lipid production, and you can indeed increase lipid quantities and find both spatial distribution, it's all at the surface, ready to be secreted, and you can quantify it, and they've done that in a number of cells. So there are many applications, biomedical, bioenergy, all sorts of applications. A little bit about disease. This is pancreatic beta cells, looking at diabetic cells. We want to look at the role of the structural changes in response to drug treatment. Again, cell heterogeneity, same dish of cells. They all look different. But what we were able to see is from this segmentation, you can segment out the mitochondria, get rid of the nucleus, get rid of the mitochondria. You're left with insulin granules. This is what's important in diabetes, the number of insulin granules and how they respond to normal person. They respond to food properly. Diabetics, they don't. If you treat them with glucose, you can see you increase the insulin granules to about 617 in this cell and 272 in this cell. So you can quantify the changes in number of insulin granules. And we've been looking at various drug treatments and see how that affects it. So again, this is a cell that a number of people down at USC are working on with Axel, trying to be able to automate the segmentation of these insulin granules and the mitochondria and the other organelles in there. So these are really complex cells. These are cultured cells. I went to a meeting yesterday on this. We have a pancreatic beta cell consortium. The normal cells from humans have hundreds of thousands of insulin granules, and we're going to be getting human cells. So we've got to be able to automatically segment all these structures in here to be able to handle these experiments. Correlated imaging, we look at, I don't have enough time to go into much detail, but we take the same cell, look at it in the fluorescence molecule, labeled, take it from the fluorescent molecule to the X-ray microscope, and you can overlay the fluorescence on the X-ray and get, you can't see it with this light, but you'll see some fluorescent molecules here. Proof of principle, we looked at a fluorescent molecule that labels silenced chromosomes. All females have 2 X chromosomes, males have 1. Females have to silence one of those X chromosomes to function normally. That's the inactive X chromosome. This is a protein that preferentially binds to that inactive X chromosome. So we could see that in the fluorescent microscope, some optical sections, those same sections in the X-ray microscope, do the overlay, and you can see where that chromosome is. You can then pull out the structure of that 3D chromosome, as you see it here, and we know that those colors reflect different degrees of crowding within that X chromosome, the dark blue being the most highly compacted of the chromosome, which is shown here. So the high compact, and that is the most highly compacted chromatin in the entire nucleus. So it's really crowded as they silence these genes. Medium lac and then the low lac. So you now get a view of the chromosome, and to my knowledge, the only view of the structure of a chromosome in interphase in an intact cell. So we can start looking at various regions of chromosomes, but that requires fluorescence. You have to have the correlated. Now that's another challenge. Overlaying the fluorescence on top of the X-ray image is not trivial, and so we need to automate that. And we're making a cryo-structured illumination microscope, so we'll get even better super resolution fluorescence imaging to try and overlay that on top of the X-ray. And that's giving Axel a few sleepless nights. But I'm convinced he'll be able to do it. Membraneless organelles. This is a really hot topic, phase transitions. For years and years, we've studied all those organelles that have membranes around them, but there are some in the cell that don't have membranes. And the most classical one is the nucleolus. So this elaborate structure, these 3 regions, The surrounding part is heterochromatin, but these three structural regions inside form simply by phase separation events. And this is important. This is where transcription occurs, that lowest absorption region, and then it moves out to the periphery. So understanding more about these phase transitions is a very interesting topic in the field of cell biology right now. And so we started doing some in vitro experiments where we could correlate fluorescence and X-ray with some poly A and poly U, poly C and poly G, and then look at those with the X-ray microscope. And you can see the different degree of phase separations with these different nucleotides. This one forms this very bizarre structure here. And if you take one of those, or actually combine the A and the C, poly A and poly C, and then just look at the 50-50 combination, this is from 100% to 0%, and inverse that all the way down. Let's just look at the 50-50, and you can see you get phase separation inside of the phase-separated blob. Now, what I'm not showing here is some of the phase separation we've done with DNA and heterochromatin, and they form some really elaborate structures, which also would benefit greatly from some automated segmentation approaches. And this is the poly G plus poly C, which is, again, a very bizarre form structure. So phase separation is something else that we're finding have elaborate structures, and you don't want to sit here and manually segment, because with the phase separation, you get thousands of droplets, and so we need to be able to automate all of these things. And I will stop and let people ask questions. This is the National Center for X-ray Tomography. We're supported by, mainly the microscope is supported by NIH and DOE, but then all of these other grants are providing money that have helped us do experiments. And Gordon and Betty Moore Foundation gave us money to build the cryo-structured illumination microscope, which Axel will have running probably in another 6 weeks. I'm over-promising. And then another program. And these are all the wonderful collaborators that bring fascinating cells to us. And so I want to thank you for your attention, and please ask questions.
Any questions for Carolyn? Just want to hear.
I love your talks.
Oh, thanks.
I'm just curious, and probably due to my ignorance, and how to do cytogenesis using these imaging instruments that you have at hand, right? Because I think part of understanding how these structures are evolving in time is to be able to get a cell, take a picture here, and then how quickly these things are changing and how you do that with the cryo.
Yeah.
My ignorance is like you do cryo and then you kill the cell.
Yeah, that's not ignorant, that's true.
Uh-huh. Oh, okay.
Yeah, you definitely kill the cell.
How are you able to do that imaging, knowing that these cells change a lot, and in the presence of pathogens, they are going to change differently? How to cope with all this variability?
We will never have the same cell over time. So what we do is fixed moments in time. So like with the neurogenesis, and neurogenesis is more easy to monitor because we have the cells and we have labels inside the cell that tell us what stage of differentiation they are. And so we can grab a cell at different stages of differentiation. We don't know the timing within that stage of differentiation, but with numbers, we're hoping to be able to get more information out of that. We're also, with the insulin granules in the pancreatic beta cells, stimulus with glucose will cause the cells to make more insulin granules. So that biogenesis is something that we're starting to study over time, because you can do time courses, 5 minutes after addition of glucose, 10 minutes, or you can even truncate that and get smaller, 1 minute, 2 minutes, 3 minutes. So you freeze cells at different time points after you trigger the event. Again, it's not the same as watching it in vivo. That's best done with something like fluorescence in a zebrafish or a mouse or chick embryo. It's really hard. It's really, really hard to get those things accurately over time. And in my opinion, the best is the model system, zebrafish and mouse, with a fluorescent microscope, monitoring things over time. We'll never be able to do that. Because, well, main reason, nobody gets out alive. X-rays kill them. They go in fresh and alive, but they get a huge radiation dose. So they're dead, and they'll never do anything again.
I see.
So it's got to be statistics.
Are you referring to the cells that you're imaging?
Pardon me?
Are you referring to the cells?
The cells that we image, none of them get out alive. Yeah. It's a really hefty radiation dose. The soft energies are much more deadly than the hard injuries, hard X-rays, because the hard X-rays just go zipping right through where the soft get absorbed.
That's great.
Do you have another question?
Yeah, let's open to
Any other questions?
Who has the solutions?
Speaking about that, then I do have a question, particularly about previous papers that you had. I mean, there's a lot of manual segmentation, a lot of curated data, and this is gold for this crowd. I mean, we could really work as part of a hackathon or even part of a thesis.
Oh.
Is there a way, a mechanism? Do you have a library in which you can make some of those available? Do we poke Axel?
We poke Axel, but there's also one thing I can tell you that Pancreatic Beta Cell Consortium would willingly include anybody. They do hackathons. They're doing periodic hackathons to try and tackle these data projects. So if anybody's interested in working on these pancreatic beta cells, I can hook you up with Kate down at USC. They'd be delighted to welcome anybody into the fold.
That's lovely.
And as far as our data, as long as the people that we've collected the data for have published it and they're ready to relinquish it, it's available.
That would be great. So certainly we're going to be poking Axel to get a few links and have these data sets shared with this community so we can try to help as much as we can.
And we do have one set of 10 cells that Jamie used for his segmentation, and some people down at USC are going to use it for theirs. So we're sending the same data set to multiple different people, which I think will help as a bit of a quality control, because then you can compare methods on the same data set. I think that's kind of a nice idea.
That's wonderful. Yeah, we want to get a hand on those data sets.
Yeah.
Please send our way.